Hello and welcome to Nikolai's genetics lessons. Today I have prepared two questions for you. One is true or false and another one multiple choice question. And here's the first statement. Bacteria are able to follow DNA instructions from a cow because the same message RNA codons code for the same amino acids in bacteria as they do in cows. And on the first glance it looks like true statement because really message RNA codons code for the same amino acids in bacteria and in cows, but still message RNA from the eukaryotes and cows belongs to the eukaryotes cannot be expressed in bacteria that is belongs to the prokaryotic group because, and here is a drawing that would explain. So imagine that this is DNA strand of the eukaryote. On this DNA strand, message RNA is coded, and message RNA looks as follows. So here we have exons and introns. And prokaryotic organism doesn't have introns, and that's why we cannot use this message RNA or this DNA fragment. We cannot just insert it into the prokaryotic organism such as bacteria, because prokaryotic organism just wouldn't be able to access these introns. So this is premature message RNA and this is mature message RNA. Of course this is continuous. I just use to show gaps in between in order to show you that here exons would be excised and this mature message RNA would be made. So how do we then express eukaryotic message RNA in prokaryotes? And we use this, or scientists use this mature message RNA and use reverse transcriptase once again to use this strand in order to build DNA strand. And now this DNA fragment, as you see, would code for the same protein but wouldn't contain introns. So now this fragment can be inserted in the bacteria and bacteria would be able to express this message RNA produced from this DNA strand. And of course this is not all the modifications that we do. We also have to take into account that prokaryotes use the same codons to code for the same proteins, but the codon usage or frequency usage of the different codons would be different. So some of the modifications also need to be done here, and then such DNA can be inserted in the DNA of the bacteria. And then bacteria would be able to produce message RNA that doesn't contain introns. So our answer would be false. Prokaryotes cannot follow directly instructions of the eukaryotes. Some modifications need to be done. So next question. When genetically engineering bacteria, what is the vector that the gene is transferred in? And here is 4 answers to choose from, and our choice would be A, plasmid. So here is a bacteria cell, and bacteria doesn't have nucleus, so DNA just freely floating into the bacteria. And also this is not the only source of the DNA in the bacteria. Bacteria also contains small circular DNA that we call plasmid, and sometimes this can be only one plasmid per bacteria, and sometimes hundreds of plasmids can be found in bacteria. So we can use this plasmid as a vector to insert the gene of our interest that would code for the certain protein of our interest, and of course here message RNA would be expressed that doesn't contain introns, and ribosome would use these messages in order to produce proteins of our interest, and this is example how insulin is made. So this is basically all for today. Thank you for attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.